The protein I chose is tissue transglutaminase 2, or TG2. This 687 amino acid long protein is found in a lot of animals, but my discussion will be focused on its role in humans. It is a ubiquitous protein found in all tissue types, and TG2 performs both enzymatic and non-enzymatic roles in the body. Its many diverse functions are because it is a protein that exists both in the cytosol and extracellular matrix. Before diving into the array of functions of TG2, let me briefly introduce the transglutaminase family. There are 9 types of transglutaminase identified in humans, of which TG2 is the most abundant and most well studied. All of the transglutaminase family of enzymes are catalysts that promote the calcium ion dependent post-translational modification of proteins. This figure shows the primary sequences of many of the transglutaminases. The conserved residues are in pink, so we see that the family of enzymes are not so different from each other. Now let's take a closer look at the secondary and tertiary structure of TG2. In TG2, there are 4 distinct domains. The topmost of these is the N-terminal beta sandwich, which contains binding sites pertinent to TG2's extracellular functions, specifically for fibronectin and integrin, aspects of the extracellular matrix that TG2 helps stabilize. There are also 2 C-terminal beta barrel domains. The yellow section of this particular figure is the catalytic core. This is the site of TG2's enzymatic functions. The main enzymatic function of TG2, as mentioned previously, is the calcium-dependent post-translational modification of proteins, notably acyl transfer reactions. A generic acyl transfer reaction is shown in this figure. This reaction is accomplished in 2 steps. First is the formation of a thioester bond with the enzyme's critical active site cysteine residue. Second, there is a transfer of the acyl intermediate to a nucleophilic substrate. This cross-linking activity, or chemical joining of 2 molecules via a covalent bond, is a hallmark of TG2 because the protein plays such a large role in the stabilization of the extracellular matrix. Let's take a look at the catalytic core that makes this reaction possible. Within the catalytic core, there is a catalytic triad that consists of the residues cysteine 277, histidine 335, and aspartate 358. The critical cysteine does the legwork of transferring the acyl group to the triad, and the triad is located in a hydrophobic tunnel, as can be seen in this figure. The reason for this may be substrate specificity, though TG2 has 130 substrates from inside and outside the cell. The enzymatic cross-linking activity of TG2 results in bonds between polyamines that are resistant to chemical and physical degradation that are incredibly important in apoptosis. When cells are dying, the cross-linking prevents toxic intracellular material from those cells leaking into the extracellular matrix, thus preventing inflammation. The regulation of TG2's enzymatic activity is super cool. As mentioned, the activity is dependent on calcium ions. Calcium 2+ upregulates the activity of TG2 by an unknown mechanism that is the topic of current research. GTP downregulates TG2 with fascinating consequences. TG2 has a unique GTP binding site in the cleft between its catalytic core and first beta barrel domain. This site allows GTP to be stabilized by hydrogen bonding by several amino acid residues. Note that several positively charged amino acids surround the negatively charged phosphate groups of GTP. The change in structure of TG2 from the binding of GTP is huge! The binding of GTP forces TG2 to go into a closed conformation, which is up to 120 angstroms shorter than its open conformation. This figure shows the difference when the cores overlapped in the closed and open conformations. The compact conformation immediately suggests why TG2's enzymatic activity is stunted, since access to the catalytic site is diminished. This large conformational change is unusual for proteins, but the weirdness of GTP binding to TG2 does not stop there. When GTP is in its bound state, TG2 forms a dimer in solution. This again further inhibits the activity of enzymatic functions of TG2 and implicates GTP as the ideal inhibitor for TG2 because while ATP can downregulate TG2's activity by similar bonding, ATP-bound TG2 forms a monomer in solution rather than the dimer seen here and here. Okay, so the enzymatic functions of TG2 are made interesting because of the mechanism of their regulation, namely the crazy conformational changes GTP-bound TG2 undergoes. But the really interesting part of TG2 comes with its non-enzymatic functions. These functions are also not dependent on calcium and arise from TG2's interactions with cell surface proteins. Here are some of the consequences of TG2's non-enzymatic roles. These include cell growth, differentiation, cell adhesion, extracellular matrix stabilization and assembly, programmed cell death, and wound healing. Many of these activities are vital to embryogenesis, tissue morphogenesis, tissue repair, tumor growth, and metastases, which gives reason to investigate TG2 in the context of disease pathology. It has been found that TG2 is implicated in 4 classes of chronic diseases. Inflammatory, chronic degenerative, malignant, and metabolic diseases have found TG2 to be malfunctioning in terms of its failure to stabilize the extracellular matrix rather than its regulatory role in apoptosis, which may be unexpected. From these classes of diseases, celiac disease is the one that most prominently involves TG2. Celiac disease is an autoimmune disease classified above in the inflammatory diseases. It has symptoms of pain upon eating gluten and causes small intestine damage. In celiac disease, the deamination of side chains of glutamine in the presence of abundant glutamine in gluten causes cross-linking of autoantigens to TG2 and other proteins, triggering an immunological response. So TG2 has a incredibly broad range of functions in the human body as listed here. The expectation then upon homozygous deletion of TG2 in mice might be an embryonic lethal phenotype. This is not the case. The knockout mice are anatomically, developmentally, and reproductively normal, showing only some glucose intolerant equivalent to maturity onset diabetes of the young. This is astonishing that a protein with such seemingly vast responsibility can be lost without fatal consequences, and encourages the study of similar proteins or even other transglutaminases in the compensation of TG2's functions. Here's a list of my works cited.